It's interesting to remember that clinical trials is a very, very new field. The modern era of clinical trials is generally thought to have started in about the middle of the 20th century. Before then, it was unusual to assign people to treatment by a random process. That was really the beginning of randomized clinical trials in about 1948, 1949. And it rapidly became the standard of understanding how medical treatments work, because you got rid of all of the confounding that you had with observational type studies, and gradually as the years went by. So I was involved starting in the early 1970s. So it was really at the very dawn of the clinical trial era, and people gradually learned what the problems were, what the pitfalls were in doing clinical trials. At the beginning, for example, if you're doing a clinical trial where mortality or some other serious outcome was what you were looking at, you were trying to see whether you were saving lives by this treatment, it was natural for the investigators to be watching the data all the time to see whether it looked like one was getting better than the other. And then if it did look like that, to be kind of wringing their hands about, well, should we continue with this trial? And very often it became more standard that as soon as the significance level, as soon as the p-value went below 0.05, people would say, oh, we have to stop now. It would be unethical to keep on going. Well, it turned out, this is something that statisticians recognized pretty early, that if you keep looking at your data over and over and over, the chances are that even if there's no difference whatsoever between the treatments, the chances get pretty high that at some point you're going to see a p-value less than 0.05, because clinical trials data are very variable as time goes on, particularly early on when there's not a lot of data in. And so we learned that you have to have a structure for deciding when to stop a trial. And we developed sequential designs and ways to look at our data so that you have to have a more extreme value early on to stop, to be really sure that it's not going to reverse itself, or as sure as you can, as sure as it's reasonable to be. You can never be 100% sure, otherwise we would never do anything or draw any conclusions, because there's always some residual uncertainty. So that's just an example. We learned about the severe biases that can occur if you throw people out of a study because they didn't comply with the treatment, or because of some other reason that you just dropped them and didn't include them in the analysis, and you can severely bias your study that way. So there were a lot of things that we were learning as we went along.